Okay, so good evening. So tonight we're going to be moving into chapter 5 of the Psychology Second Edition textbook and talking about sensation and perception. And remember, this will be uploaded to YouTube. So if you have any questions, you may chat, you may post them in the chat. If you do not want your voice broadcast on YouTube. Otherwise, if you don't care about that, then you can just unmute yourself and ask your questions. So this is kind of a cool chapter, some interesting topics that will come out of it. It kind of goes along with the previous two chapters, right? When we were studying biopsychology and then, it just went out of my head. Chapter 3, biopsychology, 4 states of consciousness, and then 5 sensation and perception. And they kind of flow together, but they are on 2 separate exams. So, all right, so let's go ahead and get started. So when we're talking about our sensory systems, right, they are responsible for providing all the information that we need in order to navigate through the world, right? So here we have a picture of a busy street scene, right? And in that scene, you see the taxi cabs. You can imagine the honking. There's a dog there. You might hear some barking. There's you're seeing all kinds of things, different colors. And all of our senses are taking in all of this information. It's going to our brain, and our brain is processing it in ways that we can understand and navigate and interact with our environment. So let's talk about what sensation is, right? So sensation is what occurs when sensory receptors detect sensory stimuli. So it's just a very fancy way of saying, for instance, if we're looking at vision, right? Your sensory receptors, your rods and your cones located on your retina are responding to light that's coming in through your pupil. For example, there's, well, here we have the different sensory systems, right? So we have vision, we have hearing. And so when you hear the word audition, that's hearing, right? So not trying out for a play, but audition is as in hearing, right? And then smell, which is olfaction, taste, which is gustation, touch, which is somatosensation. Then you have balance, which is your vestibular sense. You have your body position, which is known as this is a tongue twister, proprioception. Proprioception can be kind of hard to and fun to say once you get it. Then there's movement, which is kinesthesia, pain, which is nociception, and then temperature, which is thermoception, right? So those are all the fancy names that go along with the things we talk about regularly, right? In regular words. But I do kind of want you to be aware of these other terminology, right? And be able to recognize that. So like when you're taking your exam or when you're doing your homework, if it's multiple choice and you are choosing between vestibular, kinesthesia, nociception, or thermoception, you kind of have to know what all of those are in order to answer choose the correct answer. So, so I do kind of want you to kind of know these. So here's the other thing about sensation. So when you were reading in your book, I'm sure you read about 2 different terms, right? There's absolute threshold, and then there's the just noticeable difference, right? And they gave some examples in the book that I'm going to talk about here for just a second. So the first part of it is absolute threshold. Excuse me. And that is the minimal amount of stimulus energy that must be present, right, for the stimulus to be detected half the time, right? And so remember, the stimulus, let's just use vision for a second, right? So light coming in through your pupil, we're going to talk more about the vision system in a little bit, but light coming in through your pupil, hitting your retina, there has to be enough light to stimulate your rods and cones in order to cause action potential. So you guys will remember from chapter 3, action potential, right? That's when the neuron is stimulated enough that it actually shoots off a signal, right? So there has to be enough of that energy present in order for action potential to occur and for the stimulus to be detected. Right? So here's an example, right? So on a clear night, the most sensitive sensory cells in the eye can detect a candle flame from 30 miles away. I think, wow, that's a long distance, right? And that's pretty impressive. Let's see. The other thing is, and here's something that's also important too, is that the stimulus that causes an action potential, it can cause it without us being consciously aware of it. So, which is also interesting. And we'll talk about that too, like the wait, what moments when we talk about audition or hearing, when we get to that, talk about, wait, what? What'd you say? Because we're not, maybe we're not paying attention and we don't catch it right away, but the neurons have reacted to the sound, right? And then we may become consciously aware of it. It's kind of like I can't think of the term. It'll come up when we get there. Then there's just noticeable difference, right? And so that's the minimum difference in stimuli required to detect a change or a difference between stimuli, right? So for example, this is from the book too. It's a great example, right? So a cell phone in a dark movie theater, you are going to notice that. And it's going to be obvious because it's dark, the cell phone goes, the light comes on, and it's going to be detectable. If you're in a bright shopping mall, or you're outside, or you're in a classroom and somebody pulls out their phone, that light coming off there is probably not going to be as noticeable. The illumination of the cell phone didn't change, but the ability to detect the change in illumination is different, right? So the just notable, the J and D, or the just notable difference here would be the minimum increase in brightness required for the change to be detected. So, and of course, it's going to also depend on what your environment is like, right? All right. So we move from sensation to now perception. And perception is the way that sensory information is interpreted, how it's organized, and how do we consciously experience it, right? And there are two forms of perception that I want you guys to be aware of. Now, these can be confusing, so I don't want people mixing up these terms, right? So there's bottom-up processing. And what bottom-up processing is that's the system in which the perceptions are built from sensory input. So in other words, sensory comes in and is distributed, right? And then top-down processing is the interpretation of the sensations that's available through our knowledge, through our experiences, and what we're thinking at the time, right? So bottom up is more like automatic kind of thing. Well, actually, top down can be automatic as well, but top down really involves our thinking about it and our knowledge and experience. So the sensory information comes in, goes through different parts of the brain, and then we respond or react to it. Now, obviously, this is happening very, very, very quickly. Any questions so far regarding anything we talked about in sensation, like absolute threshold, just noticeable difference, or bottom-up processing or top-down processing? Any questions? And then there's several factors that are involved that may affect our perception. So one of them is sensory adaptation, right? Which is we no longer perceive a stimuli that is relatively constant over prolonged periods of time, right? So you go into a house, for example, and somebody is cooking bread, fresh baked bread, right? Oh, that smells so good, right? But after you've been in there for a while and visiting and talking, you really aren't perceiving the smell of that fresh baked bread as much because you've adapted to it, right? Now that doesn't mean that your olfactory senses aren't working. It just means that you're just no longer perceiving it, right? A less attractive example of that would be a cat litter box, right? So you come home, you have a cat, and maybe you're used to the litter box, but you have a guest come over. You're not noticing it as much, but your guest is going to notice it a lot more because they're not used to it, right? So sensory adaptation. Another factor is attention, right? And there's this idea of intentional blindness, and the picture that's depicted here is from a study that they did, and what they had the participants focusing on black and white figures, and because their attention was so focused on the black and white figures, the big red cross which moved across the screen didn't, or appeared on the screen, did not get noticed. So there was that inattentional blindness because they were focused on something else. 1/3 of participants that happened to, that's a high number when you think about that. Then there's motivation that may affect our perception of things, right? So sometimes we think we hear like a phone ringing. If I'm expecting a phone call and I'm in the shower, right, all of a sudden I begin to hear phone ringing, right? Because I'm waiting, I'm waiting for that call. So I'm motivated to answer that call. And then there's also the signal detection theory, which is the change in the stimulus detection as a function of our current mental state. So we may hear things or we may not hear, see, or perceive things based on what we're thinking at the time or where our mind is at, right? Then there's also beliefs, values, prejudices, and expectations. So for example, people who hold positive attitudes toward low-fat foods are more likely to rate foods with low-fat labels as tasting better than people with less positive attitudes, right? And they've actually done studies on this too. And it's interesting how our own beliefs can alter our perception of what we are experiencing. And then, of course, there's the life and cultural experiences, right? And so the example cited here is that, there was one study that found that people from Western cultures, where there is perceptual context of buildings with straight lines, were more likely to experience certain types of illusions, such as the Mueller-Lyer illusion, which will be addressed in a couple more slides. You'll see that. Than individuals from non-Western cultures, where they are more likely to see or live in rounded huts, right? So not as much kind of like straight lines there, which is kind of interesting. All right, let me go back here for a second. So any any questions on these factors that can have an impact on our perception as we're navigating the world. All right, very good. So next we're going to talk about wavelengths and waves. So the first part of it, let's talk about amplitude and wavelength. So visual and auditory stimuli occur in the form of waves. I think everyone knows that the sound of my voice right now is creating a wave, right? And the louder I talk, right? Sorry if I blew anybody's ears out. It means the higher the amplitude, right? That's just one example, right? So with amplitude, that is, and you'll see from the picture here, let me turn on my laser pointer. So when we're talking amplitude, we're talking, that is measured from the crest of the wave, and the crest are each of these tops, right? So it's crest, 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 and then there's the trough, which is the bottom of the wave. And so the amplitude is measured from the top of the crest to the trough, right? And you'll see it displayed here along the right side. And then the wavelength is measured from crest to crest. So that is the difference between amplitude and wavelength. And you'll see that here in just a second. Hold on one second, I gotta mute. Sorry, with the laser pointer, it gives me hassle, but hold on one second. I'll be right back. Apologize for that. Today's been a pretty good day regarding coughs, and now all of a sudden my lungs want to act up. So my apologies. All right, and I think I accidentally skipped ahead. Okay, so this is where we left off. So we were talking about amplitude and wavelength. So, all right. So then the next thing, which is really connected to wavelength as well, is what's known as frequency, right? So if we look at this picture here, and if we go from the left side to the right side, right, this box, let's think of this as a period of time, and these wavelengths are passing through this section in a particular period of time, right? So that's known as frequency. So that's the number of waves that pass a given point in a given time period. And it's expressed in hertz, which is the capital H, small z. So you'll see here, right? So there's 1, 2, 3, right? 4 waves. But the longer wavelengths have lower frequencies. In other words, there's not as much of, it's not a highest number of waves that pass that given point with longer wavelengths as there are with shorter wavelengths. So when we're thinking of color, reds tend to be in your longer wavelengths, and they have lower frequencies. And then the shorter wavelengths tend to be they move down to your blues and your violets, right? And they have higher frequencies. And so this is an illustration from the book that kind of demonstrates that well. And the medium length are your greens and your yellows. So if we're talking about the visible spectrum of light, this is what we're looking at. That is frequency. Continuing on, thinking of light waves, right? So now you are not going to have to know, all of these numbers for the exam. So I don't want you to have to worry about that. The point that we're talking about here is we're continuing to look at the visible spectrum. and the light waves. We're looking at wavelength, and it's in comparison with um, other items in the world that we know about, right? So as far as size wise. So there's the gamma rays, there's X-rays, there's ultraviolet rays. Then there's the visible spectrum, which is right in this area. That's what we can see here. Now there are insects that, and I don't know all the thing, but all the research on it, right? But that can see, like in the infrared, can see a greater spectrum than the human eye can see, which is kind of interesting. All right, some of this I've kind of already talked about in the perception of color. I actually forgot I had this slide here, but it's a good review, right? So remember when we were looking at that one example from the book where it had the different colors and the different wavelengths, that the longer wavelengths were your reds. Your intermediate wavelengths are like your greens, and then your shorter wavelengths are the blues and the violets, right? And so that's what wavelength and color have to do with each other. And then amplitude and color is how intense or how bright the color is, right? So larger amplitudes, in other words, the taller from crest to trough, appear brighter to us. All right, any questions before I move into sound waves? All right. So sound waves, when we talk about frequency, frequency of sound waves is equal to like the pitch, right? So high frequency is going to be a high pitch sound, and your low frequency is going to be low pitch. And then the audible range for humans is between 20 and 2000 hertz. I don't believe I asked an exam question on that either. So I'm not going to have to memorize that. And we're going to see here in just a second when we talk about amplitudes and decibels, right, which is the loudness of the waves, what can be like dangerous, right? So, so amplitude, again, remember amplitude is the measurement from crest to trough. That's going to be loudness. So if you are asking, if you want me to raise my voice or talk louder, you are actually asking me to raise my amplitude, right? So the higher the amplitude, the louder the sound. The lower the amplitude, the quieter is the sound. And then loudness is measured in decibels, which is right here, it's lowercase d followed by an uppercase B. So that's what you're looking at. So typical conversation, 60 decibels. So if we were to measure decibels right now of how I was talking, probably right around 60, maybe a little higher. Sometimes I talk loud, right? Rock concerts, that's 120 decibels. And which is interesting because the potential for hearing damage is between 80 and 130, right? And then at 130, you may actually be experiencing pain from that high of decibels. And speaking of that, right? So here's some examples, right? So loud jets. Anybody that lives like near Miramar, Marine Corps Air Station, when those jets take off, you hear them, right? So very, very high decibels. But this gives you an example, right? From 0 decibels, 20, all the way up to 140. And then the different examples, right? So a car driving by, 80 decibels, right? This looks like a quiet pond. It's kind of hard to tell from here, right? 20 decibels. Conversation, 60 decibels. Right? So just some examples from the book there. I actually kind of like this this graphic. All right, let's see. It looks like let's see. So the paper, somebody's asking about the paper. That is, I believe, oh, it's going to be July the 23rd. That is when it is due. So in about 2.5 weeks. All right. Let's go to vision. Windows to the soul, right through our eyes. Eyes are kind of cool. All right, so there are a couple of things that I am going to want you to know about the anatomy of the visual system. Right, we're going to be talking about the optic nerve. We are going to be talking about the retina. You know, the basic structure of the eye. I'm not going to be asking about the vitreous humor. Not all parts of it. We will talk about the blind spot here in a minute. Not sure how it's going to work on here, but we're going to try a little activity and see what you guys see. All right, so let's see. So let's just start off here. So light waves, I kind of used this example earlier in the lecture. Light waves transmitted, you know, they come in through the pupil across the cornea, right? Right in through the front there. And it goes back to the retina. Where it is focused on the fovea, right? Which is part of the retina. And the fovea is what contains your photoreceptors. And the photoreceptors are connected to retinal ganglion cells. And if you'll recall from the biopsychology section when we were talking about neurons and axons and things like that, that's exactly what's happening here, right? So rods and cones, they're specialized cells designed to detect light that are connected to axons, and it's that action potential that goes out through the back of the eye. And your optic nerve is actually bundles of axons, right, that are going back to the occipital lobe, transmitting that visual information for processing. And then there's the blind spot, and that is the point where there's no receptors in the eye that exists in that part of the eye. And that's simply because that is where the optic nerve is actually exiting the eyeball. So there are no receptors there. Actually, let me turn on my laser pointer. So if we look in this area, right, this is the optic nerve. The little lines here represent, you know, the axons that are all bundled together, and your retina is along the back of the eye. It has the rods and the cones. So let's see here. All right. So you should have this displayed on your screen. And I actually played with this earlier today before class, and it actually does work staring at your computer screen. Normally when we're in class, I usually have volunteer students stand up and look at it on the big screen to see this. So if you want to do this, close your right eye and stare at the X with your left eye. And then while you're doing that, slowly move your head toward the screen. And I cannot see anybody doing this. I'm not looking at anybody, so it's okay. And actually, you can turn off your camera while you do this. But just see if that worm disappears and if all you see is just the redness of the apple. At a certain point, it will disappear and then it will reappear. So if you've gone too far, but I just want people to experience that. All right, any brave volunteers want to drop a comment in the chat about their experience real quick with their blind spot? Or you may unmute yourself. All right, I'm just going to take it on faith that everybody had fun with that. Oh, wait, yay, I got a chat. Hold on one second, got to turn off the laser pointer so that my mouse will work correctly. Ah, found it about 2 feet from the screen. Awesome, yay. So that's kind of cool. There's going to be a couple other things we're going to do here for after image here in a little bit too. That's kind of cool. So, all right. Yay. Thank you, you one brave soul. All right. All right, so let's talk about the photoreceptors. So the photoreceptors, so this is what they look like here, right? So this is like the top of the neuron cells, right? So the axons would be actually traveling outside that picture. So cones, that's for photopic, also known as daytime vision. So when you hear the term photopic, when it comes to vision, we're talking about daytime vision, and that is the cones. And cones work best in bright light condition, right? So it's high acuity color information and located in the fovea. And then your rods, which are for your nighttime vision, right? And the technical term is scotopic, right? So scotopic vision is nighttime vision. And that's what you use in low light conditions. And they are highly sensitive, but it allows for low acuity vision in dim light. It's also involved in perception of movement with our peripheral vision, right? So, and it's located mainly in the periphery of the retina, right? So they are centered in different parts of the retina because of what they do. So on the exam, kind of like what I talked about with the last exam, make sure that you're reading the question carefully, right? So like psychological and physiological look really close together and then can be easily confused, right? Antagonist versus agonist can be kind of confusing and tricky. Same thing here with rods and cones. It's very easy for you to get it backwards and think you're correct, right? So just make sure that you're taking the time to kind of understand those two things and then reading the question very carefully so that you're not making an error that you ordinarily otherwise wouldn't have made. And I'm just speaking from experience that, you know, in the past, sometimes these types of questions can trip students up, right? And so I kind of want to give you that heads up. So make sure you understand the difference between rods and cones, scotopic vision, and photopic vision. All right. Any questions on the photoreceptors? All right. And then the other thing I want you to know is the optic chiasm. And that is where the optic nerve of each of the eyes merges, right? That is this, let me turn on my laser pointer. The optic chiasm is right here, right? It forms this X shaped structure that's in your brain area, and your optic nerves, right, are coming out of each eye, and they actually cross over, right? So your left eye crosses over and goes to the right occipital lobe, and your right eye crosses over and goes to the left occipital lobe. And remember, because it's divided into two halves. What else did I want to say about that? I think that's it. Any questions on that? Right. So remember, it's the X shape, and the optic nerves actually cross over each other and go to the opposite hemispheres from which they originated. Right. So this originates in the right hemisphere, goes to the left hemisphere, and this originates in the left hemisphere and goes to the right hemisphere. And then there's our visual pathways, right? So if we go back here, I'm just going to go back here for a second, right? So the information, light information, what we're seeing in the world is coming in here. And then it goes to each side. Not only is that happening, but then we also have the other visual pathways, right? So the what pathway, and then there's the where and how pathway. So the information comes in from the front, goes to the back, and then is resent out to different parts of your brain for for things like object recognition or being able to identify objects, right? So so that would be in the lower stream or the what stream, right? So the what pathway is for object recognition and identification. And then the upper stream is the where and how, which is your location in space. And we haven't talked about this for a little while, but who knows? Okay, I'm thinking everyone can see my highlighter here, and it's circling a lobe of the brain. Who knows what lobe that is? Any shout outs or chats?
Parietal.
That is correct. That is the parietal lobe. And the parietal lobe is really important in somatosensory location. It's our location in space. It's all about the, oh man, the word is escaping me. It's on the tip of my tongue, which, by the way, we will talk about in the memory section, the tip of the tongue effect. But our location in our environment, right? So it's our perceptual orientation. So you see, that's the first area that it goes to, right? Is the parietal lobe, and then ends up in the frontal lobe, right? Which is where we might interact with a particular stimulus. This is where some top-down processing might happen, where I'm like, oh, okay, this is what I'm going to do. This is where I'm going to go, right? So, all right. Good job on the parietal lobe. All right, so the book also talks about, and psychology talks about, a couple of different theories on color, right? And so there's the trichromatic theory of color vision that says that all colors can be produced by combining red, green, and blue, right? And this applies to where color vision is controlled by the three types of cones, sorry, I misspoke, in the retina. And then there is the opponent process theory that color is coded in opponent pairs. So in other words, black, white, yellow, blue, green, red, right? And that theory goes to say that some cells are excited by one of the opponent colors and then are inhibited by the other color, which is really, really an interesting theory, right? And this applies to cells in the retina. And what research has found is that both theories are true, but for different parts of the visual system. So it's just interesting how different parts of the brain, cells, right, get specialized. One of the things that I forgot to mention, I'm pretty sure I forgot to mention this when we were in the biopsychology section too, is some neurons are stimulated by straight lines. See a straight line, it's activating that neuron. Seeing certain colors, right, in your retina, those are activating certain neurons, right? All that information gets processed and put together into a picture. The brain is just amazing. It's just really cool. So let's look at some examples here of some of these. So here's another thing that I want you guys to do for a second. So you'll notice the American flag, and the American flag is actually using the opponent process theory, right? So I'm going to shut up for about 30 seconds, and I want everyone to stare at the white dot in that flag until I tell you to stop. Just stare at the white dot. I want you to continue staring at the white dot because I'm going to give you further instructions. So just listen to my words, continue to stare at the white dot. When I tell you, I would like you to avert your eyes from the white dot and look at your wall. Hopefully it's white. Look toward a white wall when I tell you to do so. And then I'd like somebody to tell me what they see. Go ahead and avert your eyes, look at the white wall.
I think you're supposed to blink rapidly, no?
Yes, yeah. Yes, I did tell you, I forgot to tell you that. All right, I got 4 people. Let's see, 5. Oh, good. Let's see what we get. Hopefully these are all people saying, I saw it, I saw it. Oh, hold on. My laser pointer's giving me hassle. Let's see. Regular colors, yes, the flag. And hopefully, when you saw the flag, hopefully you saw it in red, white, and blue. Yes, the American flag with real colors. Yes, exactly. So this is kind of a very cool demonstration of the opponent process theory. And yes, and I did forget to tell you to blink rapidly, but it does work even without that. So kind of cool. Very, very cool. All right, and then the other thing that we're going to talk about, which is also support for the opponent process theory, and I'm not going to do it here, but it's basically the same thing, right? You would stare at the dot for 30 seconds, right? 30 to 60 seconds, and then move away, and then you would also see this after image as well in its true colors. So kind of cool. All right, any questions on the color theories that we just discussed? All right, let's see here. Looking at the time. All right. So I think what we're going to do is we are going to, it's almost 7 o'clock. So we're going to take, a regular, we'll take 20-minute break. So get something to eat. I know some people rushed home and things, and then we'll come back and complete this lecture. All right, we're back from our break, and the recording is in progress. So this is kind of like where we left off. Maybe we should back up for a second, right? So we were talking about the color theories, and now we're moving into depth perception. And depth perception is basically our ability to perceive spatial relationships in 3D, which reminds me, when we were talking about the parietal lobe, it was the word spatial that was on the tip of my tongue. that I could not think of. And here it is in front of my face. So, but yes, that's our depth perception, right? And we have a couple of different clues for that, right? So first set of clues would be our binocular clues. And that's what we rely on with when we use both of our eyes, right? And so there's the idea of binocular disparity, right? So each of our eyes actually sees a slightly different view of the world. So if you ever close one eye and then open it and look, close the other eye and go back and forth, you can kind of see a little bit of a difference, right? Of the information that your eye is receiving. And then there's the monocular cues, sorry. And that's the cue that relies only on one eye, right? So that's your linear perspective. For example, like the picture that we have here. It's when two lines seem to converge. So we're looking down the road and at the end, it seems like everything comes to a point, right? And then there's the interposition, which is the partial overlap of objects, right? So those are your monocular cues. And so why are those important? Why is depth perception important? And part of it is what we're going to talk about next. So I'm going to actually show a video. Me and babies, love babies. And so there's a video that talks about the visual cliff, right? And how babies respond to depth perception. So let's go ahead and show this.
Everyone has had some kind of an experience feeling something kind of like fear of heights, and it makes perfect common sense that we shouldn't jump over the edge of a drop-off because it's dangerous, and if it's dangerous, then we should probably be afraid of it.
Probably. Exactly how and when this hypothetical fear develops makes for pretty gleeful research inside Dr. Karen Adolph's lab.
We're discovering some surprising things that are flying in the face of, how people have studied locomotion for the last 100 years.
Including what babies do and feel when they're on a ledge.
So the story about fear of heights is that there was a very, very famous classical study in developmental psychology by Gibson and Locke in 1960 that showed that human infants and infants of a number of different species will avoid crossing over an apparent drop-off on what they called a visual cliff.
As soon as the baby can move about, he can be observed in the same way as other animals.
It's a glass table, and on one side there's a checkerboard pattern surface right under the glass. The other side of the table, the pattern surface is way down on the floor. So visually, it looks like a big 3-foot drop-off. So when human babies first begin crawling, most of them will crawl over the apparent drop-off, but after several weeks of crawling experience, then they'll begin to refuse to crawl over the drop-off. And the conventional interpretation of this was that infants are avoiding the drop-off because they're afraid of heights.
She's backing away very quickly.
No, she's not interested.
She wants no part of that side.
The experiment became the shaky foundation for the myth that crawling innately teaches us to fear heights.
Babies avoid going over the drop-off because they're afraid of heights. How do we know they're afraid of heights? Because they avoid crawling over the drop-off. So as you may have noticed, the argument is circular.
As it happened, the visual cliff was a better test of an infant's depth perception, not their fear of heights. And if you look closely, you may have actually noticed that the babies don't look particularly scared.
It is a perfectly safe surface of support. So after one trial, human infants cleverly figure this out, and then they will crawl over the drop-off.
So how do you really test if a baby's scared of heights?
You can test babies at the edge of a real cliff where there's no safety glass. It's just an actual drop-off, and the drop-off can be adjustable, so it can be little or really, really big.
And why stop there? Inside Dr. Adolph's Infant Action Lab, babies are tested on a variety of apparatuses.
You can test babies at the edge of a slope, so it can be steeper, 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 crossing bridges that are wide or narrow over a surface of support, and so on. If there's no safety glass, you can test the same baby dozens and dozens of trials in the same session. And that means that you can determine for each individual infant when they think that that surface is safe and when they think it's risky within a couple of degrees of accuracy, like 2 degrees of accuracy for a slope.
Dr. Adolph has spent 25 years running tests like these, and the resulting data sets are both convincing and adorable.
Infants spend most of each trial right at the edge of the precipice. They're reaching their arm down over the drop-off. They are putting their little feet or their little hands on the bridge and taking little tiny steps at the edge. And all the while they're doing it with either neutral or positive facial expressions.
What's more surprising is how their approach changes as they learn different forms of locomotion.
So what happens is that when babies first begin crawling, they'll crawl right over the edge. Then over weeks of crawling, their responses get more and more accurate until finally they're at adult-like levels so that they can tell precisely whether a slope is safe for crawling or whether a drop-off is safe for crawling. But then the next week, when these babies stand up and face these same drop-offs, same slopes, same apparatus, now as a new walker, they walk right over the edge. So in fact, there seems to be 4 learning curves in development as babies learn to sit, learn to crawl, as they learn to cruise, and then finally as they learn to walk. So if they were afraid of heights, you should just be afraid of heights. In the rain and on a train and in a tree, you shouldn't see 4 separate learning curves.
So if babies aren't learning to be afraid of heights, what are they learning?
I think that what babies are learning over weeks of sitting, crawling, cruising, walking, is they're learning to perceive the relations between their bodies and the environment.
They're discovering what's possible and what's just a little too far.
And you can learn that without feeling any sort of fear at all. In fact, Eleanor Gibson, the person who first invented the visual cliff, wrote later in her life, as a goat is peering over the edge of a steep crag, it knows not to walk off the edge. And she said, I don't think it's feeling any emotions at all. It just knows not to go. And that's what we're seeing in babies.
Who probably know not to go. Probably. For Science Friday, I'm Luke Groskin.
All right. So how was that for a cute video? But it kind of ties into what we were talking about with depth perception. And it's also very interesting on how, they've identified 4 different learning curves depending on, what the baby is doing, right? From crawling to walking, right? So it's very cool. Any comments or questions about the visual cliff or depth perception or monocular cues? All right. Well, then here's another cute baby. You may have already guessed I like babies. And so now we're going to be talking about hearing. So here we go. We're going to talk about the anatomy of the auditory system, right? So this is how we hear. And again, there are some things that I will want you to know from this, but mainly I'm not going to be asking, about the ossicles, for instance. I will want you to kind of understand the cochlea and the basilar membrane and the cells, because that's important to understanding how audition actually works. And we'll be touching on that in the next couple of slides. So auditory transduction. Hold on one second. Looks like there might be a question. Oh, you can't see my screen. Hold on. Oops. Darn it. All right, so now we're going to be talking about auditory transduction. And that is basically how we hear, right? So sound waves, they come in and they move along the auditory canal, right? They come in through your ear, move across the auditory canal, they strike the tympanic membrane, and it causes it to vibrate. And that vibration is what causes the three ossicles, right? Those are the smallest bones in your body, in case you ever want to play trivia question with that. And it causes them to move. And this presses on the stapes of the oval window of the cochlea, right? And so that pressure back and forth causes the fluid inside the cochlea to begin to move. And what happens is so imagine the auditory hair cells, right? The sensory receptors for sound. They're like these fine little hairs that are standing up in the cochlea, and the fluid moving back and forth causes the hairs to move back and forth. That's how they get activated. And that generates the neural impulses that then travel along the auditory nerve into your brain. And then that auditory information is sent to the inferior cochleus and then to the medial, I always mispronounce this, geniculate nucleus of the thalamus, and then finally to the auditory cortex, which is in the temporal lobe. And all of that is how that gets processed, right? So that's the basic picture of that. I actually have a video that I will be posting because I just remembered that I had it and I forgot to post it. It actually is a short video and it kind of demonstrates this picture through the canal. So if we were to back up, right? So the sound comes in here, hits the membrane, presses on the ossicles here, and then this is where the hairs are located. And the fluid inside here moving back and forth. Sorry, I didn't have my laser pointer on. These are where the hair-like receptors are. And that fluid moving back and forth causes them to have action potential, right? That's how they get stimulated and send the information to the auditory cortex. So when it comes to, excuse me, pitch perception, there's a couple of different theories, right? So the first one is the temporal theory. And that theory says that frequency is coded by the activity level of the sensory neuron. But there's a problem with that. And the problem is the frequency of action potentials doesn't really account for the entire range that we're able to hear. And then because there's a point where the neuron actually cannot fire any faster, right? It reaches its max potential and can't go any further. So then the place theory, which is the next theory, is that different portions of the basilar membrane are sensitive to sounds of different frequencies, and that the base responds to higher frequencies and that the tip responds to lower frequencies. Both of the theories do explain different aspects of pitch perception. And then the rate of action potentials applies to up to about 400,000, 400,000. Sorry, I misspoke, misread. 4,000 hertz. But higher frequencies can only be encoded by using the place cues. So those are the two different theories there. Any questions on that? All right. And then how is it that we localize sounds, right? So just like with vision, right, there's monocular and binocular. Here there's monaural and binaural cues, right? So one ear versus two ears. So with one ear, each ear is interacting with the different incoming sound waves differently. And then with the two ears, provide information on the location of sound along a horizontal axis. So imagine you're standing up in that horizontal axis. And it relies on differences in the patterns of vibration. So then there's the interaural level difference, right? Sound coming in from one side of the body is more intense to the closest ear because of the attenuation of the sound wave. And then the timing difference is there's a small difference in the time at which the sound waves arrive at each ear. So those are the ways that sound localization happens. And then there's hearing loss. And your book talks about a couple of different kinds of hearing loss. So obviously deafness is the partial or the complete inability to hear, right? So there could be different levels of deafness. And then there's congenital deafness. So whenever you hear the term congenital, that means from birth. The individual, whatever it is that they're experiencing, in this case, we're talking about deafness, that means they were born with that deafness. And then there's the conductive hear loss, and this is associated with failure in the vibration of the eardrum or the movement of the ossicles. And this can be caused by age. There's you might have a person might have a genetic predisposition. There's environmental effects. Like if you ever walked by somebody who's got their earbuds in, but you can hear their music, that would be an example of an environmental impact that an individual is doing to themselves, that they may experience hearing loss later in life, right? And then, did I mention age? Yeah, age can also have that impact as well. And then there's the sensorineural hearing loss, and that's where there's a failure to transmit neural signals from the cochlea to the brain. And so there's a break at that point. And that can be caused by Meniere's disease, which causes or results in the degeneration of the inner ear structures. So those are the different types of hearing loss. Let's see here. Okay. All right, oops. That went too fast. I apologize for that. So here's an example of conductive hearing loss, right? So you see a guy here working with his jackhammer. If you've ever worked with a jackhammer, jackhammer themselves are loud enough, and then imagine jackhammering concrete. Just compounds the factor. And then there, of course, there's the picture of the Black Keys here, right? Rock musicians and concerts. Now we're going to be looking at the chemical senses. So we're talking about gustation and olfaction, which is your sense of taste and smell. And the reason why they're known as chemical senses is because the receptors actually respond to the molecules that we eat, right? So that's how we taste. And then how we smell is the molecules that are in the air that we breathe, right? So that is why it is known as the chemical senses. And so for gustation, this is what research has demonstrated, that we have 6 different groupings of taste. So we have sweet, we have salty, sour, bitter, umami, right, which is that taste for that monosodium glutamate. And then there's also research that suggests that we possess a taste for fatty content of food, right? And so, so taste buds are actually groupings of taste receptor cells that have hair-like extensions that protrude right from the central pore of the taste bud. And something very interesting there is that they have a half-life of about 10 to 14 days. And then, of course, the transduction occurs when the taste molecules bind to the receptors, causing the chemical changes within the receptor cells. And that information then is sent as neural impulses to the brain so that we actually recognize the tastes. So here's kind of a blow up of the cells, right? So here's your taste pore, and then these are the receptor cells in the bud, and then there's the axon moving out. So it's kind of a cross section there of a tongue. So who knows why taste might be important? You can type that in the chat or shout it out. Why is taste important?
Memory, perhaps, or associating certain tastes with certain memories, maybe.
Well, that's a really good guess because you are correct, right? So certain smells, certain tastes can be associated with memories. We know that emotions can be associated with that, and we will definitely be touching on that in chapter 8, right? Or 7.
Like a warning sign?
Yes, warning. That's one thing, right? So taste can help us determine whether something is safe to eat or not, right? So there's an evolutionary advantage to it. And then someone else says to encourage the process of eating and to differentiate poisonous from nutritious. Yep, you guys are getting it. That's exactly it, right? So it does have those important evolutionary life preserving factors, right? But also getting back to the memories too. Eating is one of those things that when we talked about in biopsychology and neurotransmission, that is one of the most natural ways that we can release dopamine, right? Eating is enjoyable so that we repeat the behavior, right? So very good. All right, so now we're on to smell or olfaction. And here you see this diagram. I'm going to turn on my laser pointer. Here's the olfactory bulb, right? And this is where all the nerves go back and to provide information in the brain. And then these are your olfactory receptors, right? Which are these hair-like structures or extensions that serve as the site for odor molecules, right? In order to interact and to bind with them, right? So again, just like with taste, it's chemical changes that cause the signals to be sent to the olfactory bulb where the olfactory nerve actually begins. And then I'm sure everyone has heard of pheromones, right? And pheromones are those chemical messengers that are sent by another individual. So many species, many animal species respond to pheromones sent by another individual, usually communicate information about reproductive status of a potential mate. So it's pretty obvious why that would be important, right? For continuation of the species etc. All right, and then the next section, well, first, make sure there's any questions on gustation or olfaction. I think the thing that I really want you guys to remember is that they are called the chemical senses, that they respond to molecules, and the action potential occurs as the result of chemical changes when the molecules are binding to whichever receptors we're talking about, whether it be the olfactory receptor cells or the taste receptor cells. And then there's touch. So touch has several different things going on, right? So you see this cutaway of some skin. You'll notice that, so here's the skin surface, and you'll notice there are different types of cells, and these different types of cells respond to different types of touch, right? So there's the Meissner's corpuscles, right, which respond to pressure and lower frequency vibrations. And you'll notice they tend to be, a little bit closer to the surface. And then there's the Pasolinian corpuscles that detect transient pressure or higher frequency vibrations, and they tend to be a little bit deeper in the skin. And then there's the Merkel's disc, which responds to light pressure. And again, you'll notice that it's up closer toward the skin surface. And then finally, there's the Ruffini corpuscles, which detect stretch. And it's kind of interesting. So you'll notice the shape of the Ruffini corpuscle. And so you can even imagine what that might look like when it's just detecting, stretching of the skin, right? So those are the 4 different touch sensors that are located within your skin. And they're specialized, as we've discussed. So. And then there are thermoceptions and then nociception, which is temperature perceptions, right? And then pain is nociception. This is the sensory signal that indicates that there may be potential harm coming, right? And that sends directly to the brain and through the medulla, the thalamus, and right up to the somatosensory cortex, which is located on the front side of the parietal lobe, just behind the frontal lobe. And so when it comes to nociception, right, the pain perception, there's different kinds of pain. There's the inflammatory pain, and that's signals sent from certain types of tissue damage. And then there's the neuropathic pain, which is caused by damage to neurons of either the peripheral or the central nervous system, right? So this is where you would talk about like people who experience sciatic pain, right? So that's some kind of pressure or damage to the sciatic nerve as an example, right? And then there's a rare genetic disorder that, at first thought you might think, oh, that would be cool not to have to experience pain, but not really so, right? Pain is designed to tell us that we're in danger, that there's harm, and to react to it. So with congenital insensitivity to pain, that's where an individual is not really able to detect or have the ability to feel pain. Now they can detect temperature changes, they can detect pressure, but not really experience pain reception or perception. And the reason why that's an issue is that individual is at a greater risk of injury, right? And they have shorter life expectancies. So imagine, you know, well, I'm I'm imagining someone using a saw or something, right? You'd lose your hand before you'd even realize. You might feel the pressure of that, but you would never feel the pain. So, so that is, sorry, I lost my train of thought. Yeah, so that is definitely problematic, especially to avoid serious injury, right? People have been known to like damage their tongues, things like that, because they're just not able to experience pain. All right. And next we moved to the vestibular sense, which is how we maintain our balance, right? And the major sensory organs of the vestibular system are located next to the cochlea. So here's the cochlea here. But it is also the fluid that's in the vestibular sensory organ. So, oh, I don't have my, sorry, I turned off my laser pointer. So here's the cochlea, right? And then here is the vestibular sensory organs. So, and again, just like the cochlea, these are filled with fluid as well. And they respond to, like if you're turning your head, right? They're responding to that, responds to gravity. And that's what helps us be able to maintain our balance and basically navigate the world. Any questions on vestibular? Any questions on the shoot? I hate when I lose that. On pain perception or the congenital insensitivity to pain, or any question on thermoception or nociception? Going through that. All right. Well, the next thing we're going to talk about as far connected to the vestibular system is proprioception, which is the perception of like your body's position, right? And then kinesthesia, which is the perception of your body's movement through space, right? And so the information for these travels to the brain through the spinal column. And then the information in the brain is sent to sensory organs of both of those systems, right? So that's what helps us, respond, react, move, and know where we are. All right. Any questions before we move on to Gestalt principles of perception? And then we're going to be wrapping up this, and then I'm going to start on chapter 6, so we can get some of that in now, and we'll finish up on Monday with chapter 6 and move into 7. All right, so Gestalt principles of perception. If you recall from the introductory psychology, Gestalt meaning like the whole, right? And so Gestalt psychology is that field of psychology that's based on the idea that the whole is different from the sum of its parts, and that when we're involved in things, that our brain creates a perception that is more simply than the sum of the available sensory inputs. So, and the other thing is that Gestalt psychologists say is that the brain does this in predictable ways. So their principles that they work with are the following, and we're going to kind of like talk about each one of these over the next several slides. So first, there's the figure-ground relationship, which we'll look at. Then there's proximity, similarity, continuity, and closure. And when you see these, it actually kind of makes sense. And then you'll realize that you're doing this all the time, right? So first is the figure ground relationship. So it's the idea that we tend to segment our visual world into figures and ground, right? So the focus is of the visual field, right? And the ground is the background. So and our perception can vary depending on what we view as figure and ground, right? So for example, in this concept, in this relationship here, this can be either viewed as a pair of faces, which ironically, every time I see this is the first thing I see. For whatever reason, that's what I perceive right off the bat. I don't know about you. But then, you know, when it's pointed out that it can be perceived as a vase as well, now I'm able to like really perceive the vase. But what's interesting for me, again, this is just me, is that I actually kind of have to focus for a minute to perceive the vase because I keep getting distracted by the two faces. So I'm not sure anybody else have a different experience as we're looking at this. Or maybe not.
That's
All right.
as well.
Go ahead. Go ahead.
I see the faces as well. It's really hard for me to focus on the face.
Yeah. It's like I see the faces right off the bat, and then exactly, and then I just gotta. I'm like, oh yeah, there's a vase there, and then I've got to focus on the vase. Yeah, it's kind of interesting. It's just the way my brain is working. Yeah, and we find that people perceive things differently, just kind of like what we were talking about in the very beginning of this, right? Our perceptions is based on our experiences, our available knowledge. Why my perception is the way that is, not really sure. And yes, it is an odd shaped face. Yes, it is. Yes, it is. That was a comment that came in through the chat. The next, excuse me, the next principle is the principle of proximity. And what this is that the idea is that things that are close to one another tend to be grouped together, right? So if we look at group number A, that's just one block of dots, right? You look at that, that's just a block of dots. And then if you look at the picture on the right side, right, picture B, then you see 3 columns of dots, not so much a block anymore. But we're grouping things together in that way. Even though the individual parts, that's an individual, no, I didn't turn on my laser pointer, right? So we have individual dots here, right? But when we're first looking at it, it's like, no, this is a block, right? This is a column, for example. Let's see here. Another interesting comment going back to the figure ground. If I lean back, I can see the vase. Up close, I see the faces. So again, that's another idea of those probably are monocular and binocular cues at play. And then of course, distance. Yeah, very good. All right, let's see here. Then there's also the Gestalt principle of similarity. And that's the idea that things that are alike tend to be grouped together. So hold on one second, sorry. This is the fun with doing this live and using the different tools. Sometimes it doesn't always work out the way you want, so I apologize for that. So the idea that things that are alike tend to be grouped together, right? So when you're looking at this array of dots, you are likely perceiving alternating rows of colors, right? So pale yellow, blue, etc. And then there's the Gestalt principle of continuity or good continuation. easier to say, right? And so it's the idea that we are more likely to perceive like continuous, smooth flowing lines rather than jagged, broken lines, right? So when I look at this, I'm not sure what you guys are seeing, but when I look at this, I see kind of an X, right? Two lines crossing over each other. And the other thing about this is rather than seeing this as a line, let me turn this on again. Rather than seeing four separate lines, right? One line, two lines, three lines, four lines, it's more likely that we're actually perceiving two continuous lines crossing over each other is the point of the good continuation. And then there's the Gestalt principle of closure. So as we look at this, right, my bet is you guys are instantly recognizing a circle and a rectangle, right? And in the book, and I meant to put this actually on a slide too, there's actually a sentence in the book that kind of demonstrates this, where you can read the sentence, but the words are all jumbled up, but yet you're able to recognize it. And that is another Gestalt principle that's being demonstrated. That our minds do wonderful work in helping us recognize things. So those are the Gestalt principles. And any questions on any of the Gestalt principles? And then on our final slide for this presentation, what do you see, a duck or a rabbit? First thing I always see is the duck, and then I see the rabbit because I know what it is. But even though I do this presentation and have done it multiple times, I still see the duck first. Anybody else see anything different? Anybody else see something else first? The rabbit first, or?
I've noticed it depends on where I focus my eyes. If it's more to the right side, I see
Yes.
the rabbit.
Yeah.
If I just look at it, my natural eye, I always see a duck.
Yep. Very good. Again, all about that perception. Yeah. Very good. And a lot of the other people were with me, right? Duck first. Yes. Duck first. But I do. All I see is the duck. Okay. Well, here, let me put on my laser printer here. Laser pointer. So here's what I see. I see the duck, right? So there's his beak, and then there's his eye. But if you look over here on the right side, you can begin to see the outline of the rabbit, right? So there's the rabbit. There's a little indentation where his nose and mouth might be, his eye, and then his ears flowing backwards, right? As if he's looking up. So yeah, cool. Interesting. I like getting different perspectives on how people are seeing things. So excellent. All right, so that's going to conclude chapter 5. And I'm going to end this recording here in just a second. Make sure no other questions, not seeing any. And we'll move on to chapter 6.